Hey everyone, it's Tom the Nutritionist here with Whole Life Nutrition. Super excited. We have another session of our Progressive Practitioner Coaching Program happening, and from all the feedback we've had from the previous programs, I'm super psyched to give you what they got. They got lots of protocols, lots of ideas, lots of scientific references, and many of them say they've shifted their entire practices. That's fantastic. That's what we're here for. We're here to help as many people as possible. You know, the reality is, I've been traveling around, I've been learning from these world experts in all these different fields, gastroenterology, immunology, neurology, and I'm just astounded that they found the answers to a lot of these diseases and disorders of suffering, and yet many people don't know about them. So my goal in this particular program is to get you feeling solid, to get you feeling excited in your clinical practice, in your science, in your biochemistry, in your nutritional tool chest of ideas. That's it. That's why we're here. So let's get rolling. And tonight we have a really wonderful subject. We're going to talk about GMOs. And I've had a unique circumstance in that I've been surrounded by GMO experts. Jeffrey Smith is a personal friend of mine. Dr. Don Huber, a plant pathologist I've studied with. Now, we just had Howard Vliger here in town. I got to learn from him. Pam Leary, who authored Prop 37 in California. So there's a lot of data swirling around in my head once again, and I can't wait to share it with you. So here we are today. We're talking about GMOs, but we're also introducing you to some ideas about what is health. What is the idea of dis-ease? When someone is at a state of dis-ease, usually they're not viewing this. They're not usually in a state of peace and calm like I am when I'm up in the mountains with my children hiking along this ridge line. It's called Skyline Divide. And we see this beautiful view of Mount Baker and all the wildflowers, and we're drinking from spring water and breathing immaculately clean air. It's amazing. It's really nice. And when you're away from this, you're buried in your office and you're having stress and conflict with your spouse and you're consuming junk food, it's really obvious to see the difference between health and dis-ease. So one of my friends, Dr. Stephen Jenis, published a really nice article, What's Making Us Sick? And he talks to us about nutritional status. So what we're finding is what makes us be out of our ease state, in our state of calm, is usually primarily two factors. One of which is you're getting irritated by outside substances. So environmental pollution, for example, air pollution or water pollution, atrazine, for example, herbicide in the water, or you're getting plasticizing agents like phthalates on your skin, whatnot. Something's causing irritation in your system. Combine that with a lack of nutrients, a lack of nutrition to kind of support that specific irritation. So you're overwhelmed and you can't process the information you currently have. And we begin to see how important this nutrition is, that science of that which nourishes, that which supports life. How important all these essential cofactors might be to kind of keep us from going under on all our exposures and our stress and everything else that's happening in our lives these days. There's just so much happening. So we look at ingestion, digestion, absorption, assimilation, utilization, and nutritional status. Folks, I apologize. I get so excited, right? So I stumble sometimes on my words and I speak really fast. I'll do my best. I'll take a deep breath and here we go. So the interesting piece about absorption, you'll see nutrients from non-ingested sources. So what are you getting from your air? What are you getting from your skin? So we're going to explore all this. It's a fascinating science. And we're going to look at Dr. Jenis's work, who I've interviewed, and he says, look, this is what's happening in conventional medicine. Everybody knows this one, right? I mean, they're assessing a patient, they come in, maybe you've got 7 minutes with a doc, and then they'll put you in this diagnostic criteria box, and they'll say, okay, why are you here? Oh, I see. Gas, nausea, bloating, constipation, alternating with diarrhea. Sounds like your bowel is irritated. Therefore, I'm going to give you a diagnosis of irritable bowel, and I'll give you some sort of anti-inflammatory. And it's like, okay, great. You have a name now. You went in with an irritated bowel. Now they tell you you have an irritated bowel. And they'll give you a prescription to calm the symptoms of the irritable bowel. But are they ever getting to the cause? Is there ever even a question as to what is the mechanism? Why in the world did I end up with this irritable bowel to begin with? We've got some great data. We have some amazing studies. 30% of the people presenting with irritable bowel are reacting to one specific food, and another 30% are reacting to another group of foods. So it's going to be phenomenal. You're going to get a lot out of this course. I can't wait to share it. So here's what most people are looking at. They're looking at germs. They're looking at genetics. They're looking at psychological factors. And then they're avoiding these two on the right. Everybody says, yeah, come on, Pester, we know about germs and genetics. The majority of diseases caused by genetic. Baloney. Baloney. You look at the twin studies, you're lucky if 25% of the equation is, quote unquote, predetermined because of your genetics. The rest is like all environmental. It's usually more than that is environmental. So why aren't we looking at what kind of toxins we're exposed to? The psychological thoughts? Why are we looking at the deficiencies of nutrients? We have some beautiful review articles from Bruce Ames looking at how that can shift your entire disease process. So deficiencies, toxins avoided, we'll focus on those a lot. What kind of irritants, what kind of toxins, what kind of excess are we talking about? We're talking about the environmental toxicants and reactive food particles. Folks, if you took just the clinical data that's out there right now on gluten sensitivity and you said, okay, 1% of the population has celiac disease, 0.4% has a wheat sensitivity, a wheat allergy, an IgE-mediated wheat allergy, and then you said an additional 6+% had a wheat sensitivity that's not celiac disease related. Yeah, there's a new category for that, by the way, and I'll share that research with you. And holy smokes, you're jumping up to a very large percentage of the population having a food reaction to one specific food group. We'll cover that ad nauseum, no problem. A lack of nutrients, sulfur, essential fatty acid, magnesium, zinc, calcium, all these things. Perhaps you're not noticing, like I am, how many people have sulfur insufficiencies these days. It completely and totally changes neurological behavior. It totally changes detoxification. Those two are tied hand in hand. We'll talk about that. Predisposition, of course, genetic and epigenetic. So, Tom, let's get to the subject. GMOs, genetically modified organisms. How does that apply to what you're talking about? How does that apply to getting too much of something and not enough of something else? Because, in the media, we're hearing every day, gosh, you know what? Genetically modified foods are identical to regular foods. There's nothing to worry about. There's no change in nutrition. There's no change in environmental issues. It's just a beautiful crop that we can sell across the globe. We can increase millions upon millions of hectares. We'll make tons of profits. It really puts the United States in a beautiful place in global economy. You know what? Eat them up. They're great. Okay. Well, what I'm going to say is, all right, that would be awesome. I'm a person who reviews research papers every single day. Every single day. And I have not found at all, I have not found the research to back up those particular claims. In fact, if you evaluate one specific idea and one specific idea only about this subject, wouldn't it be the fact that we are lacking any clinical human trials on the consumption of GMOs? There are no GMO tests. Humans eating GMOs. What's happening with irritable bowel? What's happening with eczema? What's happening with headaches? What's happening with chronic fatigue? None. Do we have the epidemiological studies? No, we have nothing. And yet when you look at some of the animal data, it's kind of damning. You kind of say, wait a second, pretty much across the board, you look at the rodent studies and you're like, okay, there's pituitary abnormalities, there's liver abnormalities, there's kidney abnormalities. This doesn't look good. Like, what's going on? So we're going to dive in and say, okay, well, where could the irritation be coming from? Where could the lack of nutrients be coming from that may be leading to these diseases, disorders in these specific animal studies? We can't talk about human studies because they don't exist. Even though you and I are consuming on average about 193 pounds of genetically modified foods a year, and 70+% of all processed foods are genetically modified, there are no human studies, and therefore we can't talk about them. But we can extrapolate from some of the data and say, is there a potential connection between the pandemic rise of autism, insulin resistance, type 2 diabetes, obesity, all of the things that we're experiencing in this quote-unquote modern day that may have an association to the industrial agriculture that we're exposed to? Maybe, who knows? Let's dive into the research and check it out. Let's look at it. Let's just evaluate it, and let's keep an open mind and see what we come up with. So, irritants. GMOs, genetically modified organisms, they are genetically modified primarily for two different things, and we'll share this in the next slide. You'll see it. One of which is you will insert the gene from a bacterial toxin into a plant, like corn, for example. And the advantageous property of that is when you have this Bt toxin being produced in every single cell of the plant, then the plant, when bitten by some of its quote-unquote predators, rootworm, for example, then that worm will ingest in every single cell of that plant some of this Bt toxin. Bt toxin works by pouring holes. It forms pores, in holes in the intestinal tract of insects. And then the bacterial organisms, the pathogenic organisms in the intestinal tract of the insects can then migrate and cause problems. So, this is an issue, right? This is an issue because the animal can't then, or in this case, insect, can't survive if it's getting bombarded. It's called the leaky gut, folks. We're going to talk a lot about that in this program. Anybody who tells you leaky gut doesn't exist is ignorant when it comes to recent science. Leaky gut is real, and we are now connecting it with changes in microbiome, we're changing in insulin resistance, type 2 diabetes. Well, this is a leaky gut for insects. You can give this Bt toxin, it will cause a leaky gut in the insects, and they die. They literally will be terminated because the gut is so leaky. Okay? The second one is that the genes from bacterium that were resistant to an herbicide called Roundup. So Roundup is one of the first billion-dollar herbicides on the planet. Really well sold. And the patent's about to expire in 2000. And so they reversed this idea. Let's put the resistant gene into plants. And we could package the sale of the herbicide along with the seed and continue, even though we lose our patent, continue these billion-dollar profits for an extended period of time. So they found this one slough area, this pond, that had a bunch of bacterium that were surviving even though there was a lot of Roundup. See, the makers of this Roundup were very familiar that Roundup was a very potent biocidal. It kills organisms quite readily. But they found this area where the bacterium were flourishing, and they said, oh, wow, we need a little bit of that trait on some plants, and can you imagine what would happen? People could liberally just come with aerial spraying and just shoot these herbicides everywhere. Only the weeds would die. The genetically modified crop plants would not. This is brilliant. This would be amazing. It would save so many man hours, and it would be so much easier. So this gene was inserted into the specific plant, and now we're seeing that in quite a few with the Roundup Ready. It's the soy and the canola and the cottonseed, and of course the corn. So you have these Roundup Ready crops, and is it possible then some of those herbicides could be potential irritants? And we're going to talk about how the use of a lot of these herbicides has weakened the actual plants themselves according to plant specialists, plant pathologists, and how then it's increasing the use of insecticides and fungicides, and those two things may be causing irritation in the animals, on the land, and potentially in humans as well. And then we'll talk briefly about allergenic proteins. Anytime you genetically modify something, you alter the protein structure. And if you alter the protein structure that was never ever seen before in nature, then you have to expect that you're going to create new proteins. And when you create those new proteins, you have to expect that some people's immune system will go, what is that? I've never seen that before. Partner that with the leaky gut, so you have a larger, unrecognizable protein, sneaks through the leaky gut, exposes to the immune system, you definitely have a problem. So whether or not that's happening with the GMOs, that's something we'll have to examine. Lack of nutrients, minerals, amino acids, and B12, we'll cover those specifically. So that's the data we're seeing in the animal research right now, is that there is a deficiency in minerals, amino acids, and B12. And we'll talk specifically about the mechanism of Roundup, which is to deplete essential amino acids. Okay, so what is a GMO, genetically modified organism, or genetically engineered organism, and how is it different? Well, in essence, what's going on is you're inserting the gene of a non-sexually compatible species into something else. And in doing so, you can never know, ever know exactly where that's going to go into the genome. And in fact, they can't really get it in there readily. So they have to use mosaic virus as a promoter. They have to use all sorts of different antibiotic-resistant gene markers. There has to be some sort of process of either shooting it in with some gold or electrifying it or doing a bacterial invasion. There has to be some way of getting the gene into the DNA itself. And when you do that modification, you will change how that gene is set up within the DNA, and you'll then, as a consequence, change how that DNA structure is laid out. So we've seen fish genes placed in tomatoes. The Arctic flounder, for example, was placed into a tomato. Spider genes placed into goats. You know, these different crossings that could never happen in nature alone. I've never seen a flounder mating with a tomato, thankfully. That's not something I want to imagine. So the unfortunate part of the insertion process, of course, is that when you insert, there's a possibility of creating some mutations. And then when you do the cloning, once you have the desired trait, so first they'll spray it with antibiotics, and they'll know which specific cells survived because the antibiotic resistant marker will keep that gene from dying. And so they'll say, aha, okay, or keep that organism from dying. So they'll say, ah, we have this organism that we want. Let's clone it, turn it into whatever substance we need. In the cloning process, insertion process, there are bound to be mutations. Now, this is interesting because in the functional medicine space where I come from, we talk about mutations all the time. We talk about epigenetics. We talk about what's going on with SNPs, single nucleotide polymorphisms. One single change in a nucleotide base pair, that one change in that base pair can change the reaction that comes out of that reading of that DNA. So, for example, usually you have a transcription factor, you pop open the DNA, and then you read that particular gene, and you make proteins from it, right, via RNA. So you make those specific proteins, and depending on what those base pairs were to begin with, it'll change the structure of the protein. If you change one base pair, you can alter the efficacy of the protein. You'll change the protein shape, and in the body, form equals function. So, man, if you change the shape of a protein and it doesn't work as well, anybody heard of 5-MTHFR, 5-methylene tetrahydrofolate reductase enzyme? So we know all about that SNP, right? We know that 60% of the general population, 49% hetero, 11% homozygous for the TT, C to T on the hetero, TT on the homo. Whew, that's a lot of speaking. Speaking too fast, making me choke. It's those big words, folks. So you have those specific 60% of the general population that either has a 30% reduction when they're hetero versus on the TT, the homozygous, they have a 70% reduction efficacy of that enzyme, and their folic acid never gets converted to 5-MTHF. We know increased cerebrovascular disease, cardiovascular disease. There's a lot of health associative risks with having a single mutation. The likelihood of not having mutations via this genetic modification process is nil. You can't guarantee that ever. You are likely to have mutations, and to be honest with you, no one knows what those mutations are. When they do the testing of these, they don't test for possible mutations. How do they know what's going to happen? They test for known allergens, known proteins. So this is interesting. Right away, I'm kind of wondering, GMO. If you want a little definition about this process and watch a really nice video, here's the link. I'm giving you all my links. I'm giving you all my slides. I'm giving you the MP3. I will make another video for you. I don't care. I want you to have this information. That's how I roll. I want everybody to be as informed as possible. And you know what? Question everything I say. Do the investigative work. Look at this stuff. Put it under scrutiny. I welcome you to do that. But here's a nice video at 1 minute and 47 seconds at this link that talks to you about what the process is. So you can put it under your own eye of scrutiny and say, wait a second, is this all right or is this not okay? So most common types. Of course, we talked about the pesticide, the herbicide tolerant, and the pesticide producing. So we're looking at the Bt toxin, Bacillus thuringiensis, producing proteins. And we're going to call them cryproteins because they're crystalline structure. They poke holes or pores into the intestinal tracts of the insects die. So what's going to happen when we have this particular protein around in humans? What's going to happen in the environment if every single cell of plants is producing a Bt toxin? And those plants are degrading slowly over time and ending up in the soil, and other animals might be addressing some of the silage and other insects and whatnot. What's going to happen? Have there been any studies on that? No. But, let's extrapolate. Let's think about this. So, if we look to the FDA and we say, FDA, okay, what are you telling us? Because you're supposed to be overseeing our food safety in the United States. What do you say about these specific organisms? And they'll say this particular policy on their website from the early 90s was that the agency is not aware of any information showing that foods derived by these new methods differ from any foods in a meaningful or uniform way, or that as a class, foods developed by the new techniques present any different or greater safety concern than foods developed by traditional plant breeding. Okay, for this reason, the agency does not believe that the method of development of a new plant variety, including the use of new techniques, including recombinant DNA techniques, is normally material information within the meaningful meaning of 21 U.S.C. 321 and would not usually be required to be disclosed in labeling for food. So, in essence, the FDA came out and told us, hey, these are not different than regular foods. Oh, come on, don't even worry about it. Don't label it, don't worry about it. Now, regardless of the fact that they had to be patented, regardless of the fact that this is a brand new technology, they're telling us, don't worry about it. And that's fascinating, because in 1998, there was actually a lawsuit that called on the FDA's internal memos to determine how in the world they came up with that conclusion. So how in the world do you come up with a brand new technology, play around with the design of DNA by inserting something smack dab in the middle of it, and we now know from the Genome Project, come on, you don't read single genes, you read batches of genes, and there are certain areas that communicate with other areas, and it's a very finite and intricate science that no one truly understands. So how can the FDA be so assured and say, come on, this is new, sure, we haven't tested on humans, none at all. We've never done really adequate animal testing until 1995, but these are not any different than regular. So confused, these guys put a lawsuit out and said, FDA, we're suing you because we don't buy that at all. These are scientists, these are religious experts, and they said, come on, we don't buy it. 44,000 internal memos from the FDA actually disclosed that the scientists never approved them like the policymakers did. They said, wait a second, these are not necessarily safe. Some of the quotes from these memos, you can find that here. You can see biointegrity.org at the top of the screen. The process of genetic engineering and traditional breeding are different. And according to technical experts in the agency, they lead to different risks. Oh. Some undesirable effects, such as increased levels of known naturally occurring toxicants, appearance of new, not previously identified toxicants, hello, genome, complicated, not sure where it's going to come out, increased capability of concentrating toxic substances from the environment, pesticides, heavy metals, and now since there's copious use of herbicides, pesticides, it makes sense, and undesirable alterations in levels of nutrients, Okay, so here we're talking about already the entirety of what I'm going to share with you. The level of irritants, the level of lack of nutrients, we're talking about it right here in the FDA internal memos about GMOs. The scientists said, look, these are not safe. Look, these may be more toxic. They may have novel new substances we don't know about, and they're likely going to have nutrient variations. Okay. So these may escape the breeder's attention unless genetically engineered plants are evaluated specifically for these changes. So I want you to scrutinize the data that's coming out on the, for example, No I-522 campaign in Washington State to label GMOs. They're saying, we're recognized, we know these are safe, the American Academy of Sciences and Scientific American, they all say they're safe. Pull apart those articles that they're talking about, and you show me the evaluative studies to look for unknown toxicants. How can they, who's done that? Who's looked for changes in nutrients above and beyond whatever they select for nutrients? Let's do broad spectrum nutrient analyses. Let's look at concentrations of herbicides and pesticides in the specific foods that are genetically modified. Because I don't see those studies, folks. There are occasional nutrient studies that show 1 or 2 nutrients, but there's nothing examining on a broad scale that would make me feel safe as a consumer, they would say, you've done your homework. Thank you very much. And we'll get into some safety studies here in just a little bit as well. Okay, so you dive into the animal studies and you say, wait a second. Well, this is a cell study from humans. And you say, all right, we were told that these crystal-based Bt toxin proteins that can poke holes in the intestinal tracts of insects, they're fine for humans. They're going to digest them. It's no problem. The reality is, no. There are multiple studies saying we don't necessarily digest them completely, especially for people who come into my practice. Because in my practice, I see people who have gastric acid insufficiency. I see people who have intestinal issues. I see people who have pancreatic enzyme insufficiency. Their digestive capacity is not that great. And they're assuming everybody's going to have fantastic digestion and not react to these specific toxicants. Okay, that's not going to happen in my practice. I know some people probably can't break these down. And here's this study saying, look, the toxins are capable of causing harm in human cells. And when you partner them with glyphosate, so for example, the Bt toxin varieties are usually double paired with genetic variations with the herbicide tolerant. So you usually have pesticide producing herbicide tolerant in the same plant. So you're looking at corn, for example, it has both of those traits. So not only are you going to get the toxins, but you're also going to get the increased load of that specific herbicide. So they're saying, look, this is not safe in human cells. Let's start looking at albino mice. And you say, are these toxins okay in the albino mice? No, they're not. They evaluated for hematotoxicity and genotoxicity, and they found, of course, there are problems. Induced microcytic hyperchromic anemia in mice, even at the lowest tested dose. Now, here's the problem, folks. If we're seeing negative effects in human cells, we're seeing negative effects in animals, is anybody looking to see how much of this stuff is in the human body? How much is in our food supply? How much is traveling into our systems and creating issues? No. But there was one study in Quebec that found these Cry1AB toxins in 93% of pregnant women and 80% of their offspring. Their unborn offspring had 80% positive for this GMO-associated toxin. Now, the interesting piece is the authors say, wait a second, you know, when we do dietary intakes, we don't see enough corn consumption by these people to justify these high levels. So where in the world is this coming from? And what they determined was it's likely coming from the animals and the animal products, eggs, dairy, beef, pork, chicken, that were consuming corn that had the Bt toxin in it. Isn't that fascinating? So, interestingly enough, they're like, look, this study is the first one to reveal the presence of circulating pesticides associated with genetically modified foods in women with and without pregnancy. So they're saying, gosh, this is a whole nother field of toxicology. Why in the world aren't we evaluating this more? So if this is what's going on, we should really be looking at this. The other piece that's kind of creepy about this genome alteration DNA issue is that this particular human study showed once you transgenically modify, you alter the genetic structure of a plant, you may have horizontal gene transfer with your own bacterium in your intestinal tract. Wait, let me repeat this again. Think about this. Bacterium constantly are experiencing horizontal gene transfer. Okay, they transfer genes back and forth all the time. Soil microbes are very well known to do this. The plant will give genome to the microbe will give genome to the plant. So yeah, okay. This now is shown in this particular study in 2004 that the transgenes can be transferred to the DNA in human bacterium. So the bacterial colonies can have DNA fragments from transgene plants. Now, my friend Jeffrey, I mean, he kind of goes out on a limb on this one, but let's be you know, out there and say, what if the Bt toxin gene were to get into the bacterium and the bacterium started producing Bt toxin all the time? Could that be a potential explanation for the 93% of pregnant women, 80% of their unborn offspring, having high levels of these Bt toxin? Interesting. Something to think about, something to ponder. Because when you start playing around with the balance of life itself, we honestly are so immature. We have no idea of the implications. I'll talk to you a little bit about some of the microRNA that travel from plants later on, but it's amazing. Okay, so let's look at some animal stuff again, and let's jump back in and say, wait a second, Tom, you're kind of getting me thinking, is this stuff safe? Is this safe? Like, have we looked at human studies? No, we haven't. Have we looked at animal data? And this is a really nice Greenpeace conference. You can find this link on YouTube. And this Ibora is a pig farmer. And he decided at one specific time to change his GMO feed to non-GMO feed and watch what happened to his pigs. So his pigs were sick. A lot of diarrhea, some of the piglets were dying, the litter sizes were decreasing, and he said, I wonder if it's the feed. So he fed non-GMO soya is how he pronounces it, soy to his pigs, and after 2 days, diarrhea decreased, use of antibiotics decreased, 0 died, ulcerations. So you could see he saved kroners is what that is, 250,000 kroners. I don't know what that is in the US, but he saved quite a bit of money. It's not as much as it looks like there. Litter size went up, 1 to 2 piglets. So you can see there was some beneficial changes. So is he an anomaly? Is he a person out in the middle? No. He started talking to his friends out there, and this is an egg farmer in the same video series from Greenpeace, and it shows he benefited by changing to the non-GMO soya. He had lower shell deformity, less water use, diarrhea disappeared. Again, diarrhea disappeared, stomach irritation. Now, intestinal irritation specifically. What we're seeing here is interesting because personally, come on, you can't really extrapolate. So take this with a grain of salt. But I'm seeing more and more people with irritable bowel. I'm seeing more and more people who have bacteriological imbalances. So I don't know. Is it possible that maybe something from our food supply or our industrial agricultural practices may be contributing to that? And this particular one and the animal studies is showing, yes, it looks like it does in these specific animals. I think we need more studies, mind you, but this is interesting to ponder, isn't it? Now, Howard just came to Bellingham, Washington. He was showing us some slides here. I picked his brain. He's a consultant in the agricultural industry, and he ran this particular trial, and he compared, after biopsy, the stomach inflammation of those that were fed GMO versus non-GMO, and he was finding that, yes, indeed, if you talk to vets across the globe, and he travels across the globe, and so does my friend Dr. Don Huber, the plant pathologist, and when they consult with these vets, they say, look, the animals that consume non-GMO feed are happier. They have less intestinal irritation. They're healthier. So it's interesting that we're starting to see these patterns coming up here. Now, this is the controversial study that came out that everybody says, oh, this is junk science, this is false science, whatever. The reality is the European Union first looked at this and said, but we're not so sure about it, but it just got vindicated recently in which they're saying, yeah, we support this study. Now, what this study was Professor Seralini et al., a group in France, they looked at rats, and they fed the rats these MON 810 corn, a genetically modified corn. And what they did with this genetically modified corn is they also showed the genetically modified corn and Roundup, because oftentimes if you have a Roundup resistant corn, come on, it's going to be having Roundup residues on it. And then they also put in Roundup in a fraction of what's allowed in our drinking water these days into the drinking water of these rats. And they wanted to see what happens to rats over their entire lifetime when they're exposed to GM crops, Roundup and GM crops. And interestingly enough, all 3 groups had negative reactions. We had females dying 2 to 3 times more likely than the controls and more rapidly. They developed large mammary tumors. Male had 4 times the larger tumors than developed 600 days earlier. Pituitary, liver, kidney abnormalities. These are the detoxification pathways, or in the case of pituitary, endocrinological pathways. They're essential for normal hormone function. They're essential for clearing out toxins. So interestingly enough, right? He was like, come on, look at the animals. Look at the data. It's pretty darn significant. There's no arguing here. But, immediately Like, there was a backlash. Other countries just pulled this corn off the market immediately and are evaluating all the GM data and saying, like Russia, we might want to ban this. Or the 64 other countries that have some sort of labeling or banning, they're like, yeah, we know. Come on, what are you guys doing? And yet, in the United States, in our media, we immediately attacked this study. And we said, okay, wait a second. They didn't have the right type of mice. These mice are prone to cancerous tumors. And yet, I had a 55-minute interview with Professor Seralini, which was unheard of, right? I think 22 minutes you get to see online via my Vimeo channel. But, other than misstating 162 countries versus 62 countries because his French is like so thick, his accent is so thick, and I misunderstood him, the interview was interesting because he said, look, Tom, here's what I did. I used the same rats, I used the same type controls, I used the same method, same corn in my study that Monsanto used to get this corn approved for safety in the UK. And people are tearing apart my study. So why are people tearing apart my study and they're not tearing apart Monsanto's study? And he withheld some of his statistical analysis because he said that's what Monsanto did when they got the actual corn approved in the UK. So I was like, are you kidding me? And he said, no, look, I'm using this to open up debate. I want people to start thinking. Like, if I'm seeing this in my rats, what was the difference? The primary difference, Tom, was that I did a 2-year trial, and all the safety trials done by Monsanto and other chemical-based companies on animal safety trials have all been for 90 days. He said, we did not notice statistical significance until after 120 days. So wait a second. We have 80+ thousand chemicals that are introduced into our agricultural, industrial lifestyle these days. Whether you're talking about foams, plasticizing agents, you're talking about fuels, whether you're talking about pharmaceuticals, whether you're talking about pesticides, herbicides, fungicides, whatever you're talking about, we have 80,000 plus chemicals. And he says, look, Tom, this is the first trial that looked at a lifetime study of one of the most commonly used herbicides at a dose lower than what you find in your drinking water now. And we're finding these adverse effects on these mice. So you cannot extrapolate mice to humans, but that's a cause for alarm. Well, we might not react the same way, but are we immune from reacting? Because that's what we're being told all the time. Is, oh, come on, it's a small enough amount, it's not going to have any effect at all. Nonsense. Everything has an effect. Isn't that one of the laws that we're bound to in physics? For every action, there's an equal or greater reaction. So if we're exposed to some of these things, we have to ask ourselves, what is the end result? What's happening due to this exposure? So, okay, yep. He says, look at this. The EPSPS enzyme, okay? This 5-enol pyruvyl shikimate 3-phosphate synthase. Long words again. Man, I'm glad I didn't cough on that one. Okay, so this specific enzyme is altered. They insert this enzyme, but it's not as effective as the general enzyme that's normally found in the plant. It allows for survival, but it's not as effective. And therefore, you're going to change then in the feed. Okay, let's back up a little bit. Roundup resistant. How do they do that? Roundup normally operates by affecting minerals. It chelates minerals. It's a very potent mineral chelator. It very tightly binds to cobalt, very tightly binds to manganese, and by pulling manganese out, you'll see in a future slide here, it affects an enzyme, and that enzyme can't work, and it can't produce essential amino acids, and that enzyme will then not produce downstream things such as caffeic and ferulic acid, which are known antioxidants. You can't make phenols because they are made in plants via the shikimate pathway. We'll get to that. We'll get to that. But basically what this study is showing, wait a second, not only are we having irritation that's being indicated by kidney abnormalities and liver abnormalities, as well as the pituitary, these irritations in the presence of a lack of antioxidants in their feed, because they're genetically modified, are a recipe for disaster. So it was a warning. It was saying, guys, look into this. Dig into this. Find out more. Is there possible connections? So look at just Roundup exposed to mice, and we see a drastic alteration in liver enzymes, in urea. So we're finding antioxidant, or I should say reactive oxygen species markers, the T-bars. So these things are upregulated when it comes to damage, downregulated for normal function. It's a possibility. We see in these animals clear evidence of toxicity with the exposure of Roundup, or the active ingredient of Roundup, which is glyphosate. Okay? So we can see after you get a certain threshold, the weight goes down, meaning they're sick. So this stuff is being sprayed all over the globe right now. Glyphosate, now it's not under patent by Monsanto anymore. So it's glyphosate-based herbicides. You'll see Scott's Weed Killer. I see my neighbors all the time out spraying their weeds in their lawn. Boom. What is this substance? It is a potent mineral chelator. It binds to minerals, and we talked about magnesium, we talked about manganese, cobalt, magnesium, zinc, iron, calcium. It binds to those minerals, making them unavailable to the plant. Now, on the websites, they'll say, oh, there's an immediate biodegradation by microorganisms and whatnot. You know what? Depending on the soil conditions, you may have Roundup around in your soil for years. Years. And not only is it a potent mineral chelator, what did we also say it was? How did they discover it? There was a pond of organisms that were resistant. Normally, Roundup liberally kills beneficial microorganisms. So it's a biocidal, it's patented as a biocidal, it's biocidal, and it's a mineral chelator. So you kill organisms and you bind minerals. If you kill off beneficial organisms, you allow pathogenic organisms to flourish. And we'll look at some research to show that's exactly what Roundup does, or glyphosate does. And we'll also see that the minerals are low. So what do you need in your immune system? Superoxide dismutase, is that a manganese-dependent enzyme? Absolutely. Okay. And what happens if you throw off your microbiome? We'll talk about that. Did you know scientists are now calling the immune system the microbial interaction system? Come on, man. We evolved on this planet in the presence of bacteria, fungus, archaea. We've been here as guests. Really, come on. They outnumber us 10 to 1. You know that, right? I mean, there's 150 times more gene expression on your body from your organisms than there are from you. So if we start throwing off the balance of minerals and organisms, are we going to have a problem? Let's look at that. Because what's happened is we've been spraying this stuff tremendously, increasingly over time, increased 6,504% from 1991 to 2010. And look at the type of weeds. We've had more weed resistant because nature is wise. Plants are wise. They will adapt. And what's happened is now we have these resistant superweeds. And we used to have just a few. Now we're getting up to 60 plus superweeds in the Midwest. Over 50% of the crops now are overrun with superweeds. So now the farmers are forced to use not only greater amounts of the herbicide, but other types. 2,4-D, part of the Agent Orange combination, as well as something called dicamba, which is a herbicide. It's very potent. It volatilizes. If you spray it on and the conditions are right, it might volatilize into the air, be carried by some wind, and settle down on somebody else's crop and kill it. So there are other alternatives, yes, and they're working on dicamba tolerant crops, as well as 2,4-D tolerant crops as a potential solution. But holy smokes, you have to start thinking about how many more chemicals is that going to use? And when you do analysis, and they've done that in this Environmental Sciences Europe 2012, they said, look, sure, BT crops have reduced pesticide use. That's great. The insecticide use itself. So you want to kill insects, the BT crops have done that effectively. But the 527 million pound increase of Roundup or glyphosate-based herbicides versus the reduction of 123 million pounds of the insecticide still equates out to a 404 million pound increase of chemical use because of genetically modified crops here. So that's fascinating. And what we're going to see later on in the slides is now the insects are resisting the Bt. So now if the Bt resistance happens in insects like rootworm, for example, now not only are you using the herbicide, the Roundup, but now you have to use an insecticide. And what you're going to see here shortly is the way these things work is they weaken the immune system of the plant. If you weaken the immune system of the plant, you're going to see more disease, fungal overgrowth, something called Goss's wilt occurs. Oh my gosh. Then you have fungicide use increase. There was a Motley Crue email that just went out the other day saying, hey, you may want to start investing in some of these fungicides because the plants are all becoming so weak that the farmers need more fungicides. So we're on a pesticide roller coaster right now. The use of more chemicals is leading to the use of more chemicals, leading to the use of more chemicals. And by the way, if you take soil and you saturate it with herbicide, and you try and plant in a non-herbicide tolerant plant, that, over time, I'm wondering if that would be more difficult to grow. Contrary to often repeated claims that today's genetically engineered crops have and are reducing pesticide use, the spread of glyphosate-resistant weeds and herbicide-resistant weed management system has brought about a substantial increase or substantial increases in the number and volume of herbicides applied. If new genetically engineered forms of corn and